Biology 1408, Chapter 2, Chemistry of Life, from the Concepts and Connections textbook by OpenStax. This chapter will cover topics which integrate chemistry into the biological world. By the end of this chapter, you'll be able to describe the chemical concepts of matter and bonding, the important properties of water, and the four major classifications of biological molecules, which will be present in almost every major concept studied throughout the rest of the semester. Let's begin by studying the building blocks of molecules. When learning about how biological processes function, it is vital to use the principles and tenets mostly associated with chemistry. In order to do this, there are some basic terms which need to be mastered, and you've probably seen or heard these terms since you were in first grade, and that's because they are foundations of studying both chemistry and biology. Firstly, the term matter. It is defined as an entity in which occupies space and has mass. We generally study matter in the forms of elements, and elements are substances that cannot be broken down chemically into another substance. Some examples of substances in elemental form are lead with the chemical formula Pb, iron with the chemical formula Fe, oxygen gas with the chemical formula O2, and the two subscripted behind the oxygen element symbol denotes that there are two oxygen atoms present in that substance. This is considered an element still because both atoms are the same element, oxygen. And one of the many elemental forms of phosphorus with the chemical formula P4. Compounds, however, are formed when two or more different types of elements are grouped together in a substance. A few examples of compounds are shown: water with the formula H2O, carbon dioxide with the formula CO2, and aspirin with the formula C9H8O4. Your textbook doesn't discuss the term compound, but I feel it's important to make this distinction. All elements and compounds are composed of atoms, and atoms are the smallest component of an element that also retains that element's chemical property. Atoms can, however, be subdivided into smaller components called the proton, neutron, and electron. There are smaller subcategories, but we will not concern ourselves with these in this course. The proton is positively charged and located within the nucleus or center of the atom. The neutron is neutrally charged and located within the nucleus. Electrons are negatively charged, located outside the nucleus. Every element has a distinct number of protons, neutrons, and electrons, and these values and other properties are used to design the periodic table that we know today. The periodic table and the information enclosed are used to determine information about each element. Elements grouped in the same column, called families, have similar chemical behaviors, and elements within the same row, called periods, have their outermost electrons in the same energy levels. When looking at an element on the periodic table, traditionally there are four pieces of information. On the top left corner of the symbol here, there is a whole number that is called the atomic number. This indicates the number of protons within that element. For the hydrogen, this value is 1. The second piece of information is the symbol for the element. This is a 1 to 3 letter abbreviation for an element's name and is used in chemical formulas. Note that the first letter is capitalized, but any additional letters are always lowercase. In hydrogen's case, the symbol for hydrogen is H. Below the symbol, a number which is typically not a whole number indicates the relative atomic mass of a single atom of that element. For hydrogen, that is 1.01. And the last piece of information is the complete name of the element. The atomic number and atomic mass can both be used to determine the number of protons, neutrons, and electrons in an atom. For example, sodium's atomic number is 11, and its atomic mass is 23. The number of protons is the same as the atomic number. So since sodium's atomic number is 11, the number of protons it has is 11. The number of neutrons is calculated by subtracting the number of protons, or atomic number, from the atomic mass. That is 23 minus 11, which equals 12. So there are 12 neutrons within sodium. The number of electrons can be calculated very simply by looking at the atomic number and determining if the atom in question is charged at all. So since I gave you a neutrally charged atom of sodium, the number of electrons has to equal the number of protons, so that would be 11. If I had said sodium was a plus charged atom, the atomic number and atomic mass are still 11 and 23. However, the electrons for this particular atom would be 10 because it is missing one electron, therefore positive more than the normal neutral sodium atom. Now, many of the elements have forms called isotopes, which have the same number of protons but differing number of neutrons. We're not going to concern ourselves with how to calculate that, but you do need to know what an isotope is and what we might use that for. It turns out some isotopes are unstable and decay into more stable elements. These are called radioactive isotopes. An example of a radioactive isotope Interestingly, radioactive isotopes can be used to determine the age of various items. For instance, the radioisotope C14 decays into N14, and it takes approximately 5,730 years for half of an amount of C14 to decay into N14. This is defined as the half-life of C14. This can be used to extrapolate the age of skeletal remains like the ones of King Richard III shown in the image. The next level chemists study is how atoms group together to form molecules. This is done through chemical bonding, and there are several types of bonding or interactions that can occur between atoms. We are only going to discuss 4 of these types. The first 2 of these types of bonding involve the interaction of electrons between 2 atoms. The first being ionic bonding, and this involves the transfer of 1 or more electrons between 2 atoms. And this typically occurs between one atom is in metal form and the other atom is in non-metal form of an element. The second type is covalent bonding. This involves the sharing of 2 or more electrons between 2 atoms, and this typically occurs when both atoms are nonmetals. When electrons are shared between atoms, they can be unequally or equally shared. Because of this property, we can further classify covalent bonding as either polar or nonpolar, respectively. The last 2 types of interactions are different because they don't directly involve transferring of or sharing of electrons. Instead, they involve interactions between atoms through relative charges or temporary induced charges. For example, hydrogen bonding is actually an interaction between a positively charged hydrogen atom in one molecule and a negatively charged oxygen, nitrogen, or fluorine of a whole other molecule. This is very important because this type of interaction is used by many biological molecules, which we will discuss later on in this chapter. The last interaction is the van der Waals interactions, and these are very weak interactions between molecules, and these only occur and they're very temporary because what occurs is induced charges within an element or atom within a molecule because of movements of electrons around the atom. And so they're temporary, they don't stay, and these interactions are very weak. I can show you some examples of these. So we have some examples of interactions and bonding. Shown here are examples of ionic bonding, where you have a sodium and chlorine, where the sodium transfers 1 electron to chlorine to create a sodium chloride molecule or what we refer to as table salt. You also have a covalent bond occurring between a hydrogen and oxygen in water, H2O, and this creates a partially positive and partially negative charge within the molecule itself that you can see that we denote here. And then you have nonpolar covalent bonding between two oxygen atoms to the right-hand side. And since they share the electrons equally, there is no relative charges within the molecule itself. One of the first types of interactions, the hydrogen bonding, occurs when you have a hydrogen atom in one water molecule. So this is one water molecule here, and you have a hydrogen right here that has a partial positive charge. And then an oxygen within a whole other water molecule has a partial negative charge, and these two interact in a form that we call hydrogen bonding, and we typically draw that as a dashed line. Now, this property makes water an excellent solvent, and this property allows water the ability to attract to other molecules. Van der Waals interactions between atoms on the pads in the digits of a gecko can actually have a positive attraction to the surface of whatever the gecko is climbing on, in this case, a piece of glass. One of the most important molecules for life is water. Because hydrogen and oxygen create a polar covalent bond, the molecule overall is considered a polar molecule. This property makes water an excellent solvent. It allows it to the ability to attract to other molecules. It also has a very strong cohesive property, which means it can attract to other water molecules through the hydrogen bonding. And it has strong adhesive properties, which means it can attract to other molecules other than water. Both the cohesive and adhesive properties give water the ability to perform functions vital to life, like traveling against gravity from the root to the leaves of a plant, and dissolving most molecules used in biological mechanisms. However, there are situations for which a biological function would need a molecule to not dissolve in water. We categorize molecules that dissolve in water as hydrophilic, and molecules that do not dissolve in water as hydrophobic. It is important to remember these terms as we will revisit them in later chapters. When substances do dissolve in water, we call this a solution. There are three types of solutions which can result: acids, bases, and buffers. Scientists use a numerical scale called the pH scale to determine if solutions are acids or bases. The scale ranges from anywhere from 0 to 14, where solutions below 7, like say orange juice, are considered acidic, and solutions above 7, like bleach, are considered basic. A buffer, however, could be acidic or basic. Its job is to just prevent the solution pH from drastically changing when substances are added to the solution. An example of a buffer is your blood. The blood is buffered to a pH of approximately 7.4. And if the pH were drastically increased or decreased, many of the biological functions occurring in you right now would cease to occur, causing death. Now we are going to study 4 different types of biological molecules. These 4 types of biological molecules perform functions that are vital to living organisms, and these molecules, sometimes called macromolecules because they can end up quite large in most cases, are categorized as carbohydrates, lipids, proteins, and nucleic acids. We're going to learn about each one of these biological molecules in more detail. The first being carbohydrates. Carbohydrates provide energy for the body, whether the organism consumes them by eating or the organism makes them themselves. They still provide energy, or still need energy, and they do this through the use of carbohydrates. The chemical formula for most carbohydrates follows the general rule of C1H2O1. So when I write a formula where there is no subscript behind a chemical element symbol, we're indicating that that means 1. So another way to write this is C1H2O1. And we put that in parentheses with an N value behind that. When you substitute any number in the N value and multiply that number by the subscripts within the formula, you can determine the actual formula for a complete carbohydrate. So if N equaled 6 and you multiplied 6 times each one of these subscripts within the general carbohydrate formula, then you would end up with something like this, C6H12O6, the formula for the carbohydrate glucose, which is a simple sugar. Now, carbohydrates can be classified into 3 subtypes based on the number of simple sugars present in the molecule. Monosaccharides only have 1 sugar molecule, and we call that a monomer. Disaccharides have 2 sugars, and we call that a dimer. And polysaccharides have many sugar molecules, and we call those polymers. Examples of all of those, a monosaccharide could be the example of glucose, a disaccharide would be lactose, and several polysaccharides I have shown here, like glycogen, starch, cellulose, or chitin, are examples of polysaccharides. I'll show you some pictures of these, where you can see glucose here on the left-hand side. It's actually in linear form, but it's 1 sugar, and if you count, there are 6 carbons, 12 hydrogens, and 6 oxygens present. Oh, let me erase that real quick. In the disaccharide lactose, there are 2 sugars, and these are denoted where you have 1 here attached to another one here. And anytime you move beyond the monosaccharide into disaccharide or above, we typically draw the sugars as ringed entities instead of linear. So you'll see the ring that's a hexagon here. And then the polysaccharide that I'm showing you here is an example of a very short molecule called starch. And starch can have anywhere from hundreds, if not thousands, of different sugars attached into that molecule. And in fact, we can look at some examples of those. Shown here are some examples of the polysaccharides that I had shown you before in a previous slide. The difference or distinction between these polysaccharides are which organism makes them and what their ultimate function happens to be. Glycogen is actually a highly branched polymer of glucose molecules, and it's stored in the liver and the muscle. It can be broken down into single glucose molecules and used to make energy when you need it. Typically, only animals make this, and are something that is highly utilized to provide energy for that animal. Starch, the polymer we had seen just before, is typically made by plants. It's also a polymer of glucose molecules, but it's made and stored, and it's only really used for energy, but it's not quite as highly branched as the glycogen that you see in animals. But plants make another type of polymer that we call cellulose, and cellulose is highly patterned into a structure. The reason the patterning is present is so that it provides stability and support for the plant structure itself. And the last one is this molecule called chitin, and chitin is made by several different types of organisms. including insects, crustaceans, and fungi. And they do this to form the exoskeleton or any kind of support structures that are used for support or even protection for that particular organism. And it looks very similar to if you look at the chemical formula for glycogen or starch. However, there are some other groups added called N-acetyl groups attached to each one of the sugars themselves to give them as different properties. Now, when we make the polysaccharide of a carbohydrate, there are a couple of chemical reactions that must occur. They're formed through a chemical reaction called a dehydration synthesis reaction, where an OH from one glucose molecule and a hydrogen from another glucose molecule are actually removed from the two molecules, and then what's left over attaches. So you create this bond with an oxygen here in between. The water molecule comes off, hence the term dehydration. The reverse can be done to separate the two sugar molecules by adding the hydrogen or water molecule back in. So you take this water molecule, add it back in, and break that bond, and you put the OH and H back in their respective spots. This is called a hydrolysis reaction. Both of these reactions require energy and enzymes to perform the function, and both of these reactions are performed in normal metabolic processes in living organisms. The second type of biological molecule are called lipids. Lipids are used for a variety of functions, including long-term energy storage, protein modification, hormones, and building of plasma membrane. Lipids are overall nonpolar, which does not allow them to dissolve in water, and therefore they're considered hydrophobic. And there are three subtypes of lipids. The first subtypes are called fats. They're made up of a chemical compound called glycerol and another compound called fatty acids. The second subtype are called lipids, phospholipids, which assemble into forming a very large structure called the plasma membrane of any cell. And then the last subtype is called steroid. And steroids have very distinct structures. They're made out of 4 rings of carbon atoms and perform a variety of functions. Some examples of these are drawn here. Fats have a classic structure with the glycerol on the left-hand side that you see here. This entire structure is called the glycerol. And on the right-hand side, 3 chains of carbon-hydrogen connections are formed in this zigzag pattern that you see here. And there are 3 of them. We typically call this, one particular example of a fat is called a triglyceride. Now, phospholipids have a similar structure to fats with a few differences. There is a phosphate group attached to the glycerol, so remember this is the glycerol structure. You also have a phosphate group attached to one component of the glycerol. And then there are these chains of carbons and hydrogens that zigzag, but there are only 2. And one of the 2 actually kinks out to the side. Now, this is actually quite difficult to draw in the larger component that it typically makes called the plasma membrane. So more often than not, we typically draw it as a sphere with 2 of these little tails that you see coming off of that. And these aggregate together to form 2 layers with the tails pointing towards one another between the 2 layers. And this structure itself is either coined the phospholipid bilayer, or we call it the plasma membrane. Now, steroids have a classic structure again with the 4 different rings that you see here, 1, 2, 3, 4. And the components that come off of the rings, or functional groups as they're called, change from steroid to steroid. The one that you're seeing here is called cholesterol. Some other examples of steroids like testosterone and estrogen have a very similar structure. The third major type of biological molecule are proteins. And proteins have many functions, from enzymatic, structural, all the way to storage. From a protein, or for a protein to be functional, it must first be built and folded into the correct structure, and there are several stages to this process. They are coined as primary, secondary, tertiary, and quaternary. And in order to discuss any of these stages, we must first learn about the building blocks of a protein, called the amino acid. And the amino acid it's a simple structure or simple molecule which has a fundamental structure that you see here. There's a central carbon atom that you have right here in the center, which then has 4 different components connected to that central carbon atom. One of them being an amino group, a hydrogen, a carboxyl group, and what we call an R group. All amino acids have the same first 3 components. That's the amino, hydrogen, and carboxyl groups. And the R group changes from amino acid to amino acid. So this varies from every amino acid. There is a total of 20 different amino acids, therefore 20 different R groups. Let me go ahead and show you an example of all of these different amino acid R groups. The diagram shows the structures of all 20 different amino acids and some basic characteristics of each. Now, you do not need to know the structure of each amino acid R group, but you do need to be able to recognize and identify components of the fundamental structure that I previously showed you in the past slide. But some interesting characteristics that you can see here are that a lot of the amino acids that you see here can be categorized as whether or not they're acidic, they're basic, are they aromatic, do they contain sulfur? So there's quite a few characteristics about each one of them, and we can actually classify them or name them through a code. And we either use a 3-letter code here that you see. So for alanine, ALA it's a 3-letter code, or a single letter code. And alanine single letter code is just capital A. Every single one of the 20 amino acids has their own 3-letter code and single letter code, which we use to build amino acids in the laboratory. So now that you know what an amino acid looks like, let's look at how we fold them and make them functional. So let's actually look at the primary structure. The primary structure is formed first by attaching amino acids one after another into a long chain, right, that you can see here. So each one of these, so this would be like amino acid number 1, 2, 3, 4, all the way to the very last one that I'll call N. This entire sequence of 1 all the way through N is the amino acid sequence that is special to a particular protein. No protein has the same amino acid sequence as another protein. Once the finished chain of amino acids then is formed, secondary structures called an alpha helix or a beta pleated sheet can occur within the chain of amino acids, and these are done by using hydrogen bonding. And so the alpha helix is a section that turns like a helix, and the beta sheet is kind of this zigzaggy little thing, kind of like what you would see in corrugated cardboard. Once the beta sheets and alpha helices have been created, the protein will fold into a 3D structure specific to the protein's sequence of amino acids called the tertiary structure. It's 3D. Every single amino acid has an important job, and the folding is what creates a functional component to the protein itself. A lot of proteins at this point are functional and can go do a job, whatever that happens to be, but sometimes it turns out that some proteins need an additional stage. And this fourth stage is called the quaternary or quaternary structure. This occurs when two or more separate proteins in their tertiary folded state associate together to form one protein complex. And what you can see here is that this light blue colored is one protein and the darker blue colored is another protein. And these two have now associated together. Only when they are associated are they then considered functional. An example of a quaternary or quaternary folded protein is a protein complex called hemoglobin. Hemoglobin is the protein within red blood cells, which carries and binds to oxygen gas and carries it throughout the bloodstream to cells in the body. Now, hemoglobin is a protein complex. It has 4 different tertiary protein folded chains, each of which has bonded to a what we call heme group. And the heme group is this large structure that you see here that has an iron 2+ atom within that. And oxygen, O2, binds to a single heme group. Hemoglobin is not fully functional unless all of these components are in place, and you can see that there are 1, 2, 3, 4 different heme groups, so this particular hemoglobin can bind to 4 O2 or oxygen gas molecules. The last biological molecule, called nucleic acids, carries genetic material in cells, and this information is what drives the overall function of the cell. There are 2 types of nucleic acids called deoxyribonucleic acid, or DNA, and ribonucleic acid, or RNA. Now, both DNA and RNA are built with smaller molecules called nucleotides. Nucleotides contain 3 main components: a phosphate group, shown here, a ribose sugar, and a nitrogenous base. There are 5 naturally occurring nitrogenous bases, and they are called adenine, guanine, cytosine, thymine, and uracil. Scientists abbreviate the nitrogenous bases to the single letters A, T, G, C, and U, the first letter of each name for the nitrogenous base. Now, in the nucleotide for the DNA molecule, the only nitrogenous bases present are the A, T, G, and C. For RNA, the nitrogenous bases that can be in a nucleotide are A, U, G, and C. Additionally, the ribose sugar has an additional OH functional group attached to this particular carbon on the sugar, giving it some special properties that we'll discuss here in just a second. Given the structures of nucleotides for DNA and RNA, each have distinct overall functions. DNA is double-stranded and turns in a helical fashion with the nitrogenous bases pointed inward, and bonded together with hydrogen bonds, with the adenine and thymine across from one another and the guanine and cytosine across from one another, that are color-coded using the red, green, yellow, and blue colors. So you can see that if adenine and thymine are always attached to one another, you will only see red and blue within the DNA molecule, which you can see 2 here also. The cytosine and guanine are also the green and yellow, so you can see the green and yellow attached to one another. RNA, however, is a single-stranded molecule and turns in a helical fashion. And the color coding is the same for adenine, guanine, and cytosine. However, uracil is the purple color. So you'll see uracil here, but no thymine blue color. The double-stranded nature of DNA and the hydrogen bonding between the nitrogenous bases is very important for many biological functions, which we'll learn more about in future chapters. Therefore, it is prudent we show in detail the bonding which occurs between each nitrogenous base. Thymine and adenine have 2 hydrogen bonds shown between them in the dashed line here and dashed line here. And the guanine and cytosine have 3 hydrogen bonds, shown 1 here, here, and here. Most importantly, the phosphate groups at the ends of each strand are referred to as the 5' ends. And the OH on the ribose of each strand are referred to as the 3' ends. This allows for indicating directionality within the DNA molecule itself. This brings us to the conclusion of this chapter. If you have been paying attention, you should now be able to describe matter and bonds, properties of water, and the 4 major biological molecules and their functions. Now that we have looked at all of the material in Chapter 2, there are a few things that you could do next to help you succeed in learning this material. Take a look at the list shown and do as many as you can. I also think it's important for you as a learner to see the same content but in a different context. Therefore, I've added a few web links which I think will be helpful resources during the process of learning all of the material in this chapter. Thank you for listening and can't wait to see you in the next chapter.